Importance of proteins. Today we will study dietary functions, building materials for animal body tissues. That is, what are the dietary functions of the proteins, why it is necessary, and what is their use in our life. First is, it is a building material for any animal body tissues, maintain normal growth, reproduction and physiological activities. It also repairs the synthesis of new tissues. It is also involved in the various enzymes, hormones and vitamins, and it is also having properties of like buffering. So the pH, body fluid, blood and cell cytoplasm. So this is all about the dietary functions. As I said, it is involved in the building materials for animal body tissues, maintain normal growth, reproduction, and it is also involved in many physiological activities. Also, it repairs and synthesis of new tissues required. It is also important in the various synthesis of enzymes, hormones and vitamins too. All enzymes are protein, but all proteins are not enzymes. That is one of the important functions of the proteins. Now we will see the various functions. That is, generally it is major division in that functions of protein. You will find that enzyme catalyst, enzyme work as a catalyst, and another is defense. Defense means antibody proteins and other proteins are to block. Then transport protein, haemoglobin, myoglobins and transferrin, these are also one of the examples of protein and their functions as a transport protein. Now certain our body is made up of certain keratin, fibrin, collagens, and all are involved in the support of the bodies. As we know that we are moving towards, and motion is there in our body and walking with that. So for that we require actin and myosin and cytoskeletons of the fibers for moving this our hands and legs. Another example or most important functions is regulation. Some hormones, some regulatory proteins on the DNA and cell receptors are also made up of that. And then storage protein, as we know that storage is found in the many plant seeds, and human beings and animal tissues all are made up of proteins, and that having storage functions too. So I will give you certain examples of proteins that having, for example, you will find venom, snake venom, that is also made up of then peacock having these are the features that will show you how the protein is useful in our bodies, and example is already being given to you. Now the biological and physiological functions of the proteins. So biological and physiological functions of protein. First is biologicals. Biologicals means in our body. Physiological is also in our body. But living organisms have both functions. So in enzyme, all enzymes are protein. Please note that all protein are not enzyme, as I said previously. That is play important role in the metabolic process. For example, ribonuclease, trypsin, phosphofructokinase, alcohol dehydrogenase, catalase, these are the various enzymes which have specific functions in the various metabolic pathways. PFK, that is in glycolysis. Then structural proteins. Structural proteins, that it is hard in structure, you will find in our hair. Okay, that is structural component of keratin, elastin, collagen, which provide mechanical support and protective layer to your hair. Okay, then transport protein. Transport, as I said, hemoglobin is the one of the example of that, and some elements and compounds are transported from one tissue to the another tissues, and these will be possible through the transport protein. Now respiratory functions, they play important role in respiratory function, that is myoglobin, hemoglobin and cytochromes, and that is there. Now nucleoprotein, it is also play important role in the hereditary functions and protein synthesis. Histone protein, that is found in the DNA attached with that is also nucleoproteins. Okay, so this is all about the functions of proteins. Now the storage protein, whose biological function is to provide reserve and essential nutrient, are called storage protein. What mean by storage protein? That is protein whose biological function is to provide reserve of essential nutrient. As and when required, it gives you. But how we are taking this? For example, milk we drink, that having a casein proteins. Many people are eating eggs, that having a ovalbumin proteins, and various oilseeds and proteins forms the most of the chickpeas, and cereals having also protein. For example, cereals maize having a Zein protein, rice having a origin protein. Okay, so these are the example of storage protein found in the various seed materials too. Now another example, biological function is a contractile or motile protein. Certain protein have a unique capability of movement. So as I said previously, that is actin and myosin, and in during the cell division phase, that mitotic and spindle of cell division. Okay, that is that. Then regulatory protein or regulation of all metabolic process require hormones, which are made up of protein, insulin, lexapron, repressors. Okay, protective or explosive proteins are also found that have a biological active role in the cell defense protection. For example, blood clotting proteins, thrombin and the fibrinogens, which prevent loss of blood when circulation system is damaged. Then it is also required in certain immunity. Immunity means antibodies. Antibodies produced when the antigens enter into our body, so it protect against the disease. Then antibiotics. Many antibiotics are made up of protein, which are reused in medical therapy. For example, gramicidin, valinomycin. Certain vaccine. Yes, vaccine is also made up of proteins. Diagnosis of disease, that is through various tests. For example, kidney disease can be detected by protein test. These are the various functions of biological and physiological functions with test too. Now, structure of histone proteins. Here is the structure of histone octamers. Various colors gives you how many octa. Octa means 8. 8 mers is found in, and that is attached with the DNA. Yes, histone protein. The chromosome, where it is localized P arm and Q arms, where these loops is solenoids and nucleosomes. So the histone is attached with that. Now the base pair and DNA is a long chain of DNA, that is ATGC, that will form the genes. So this base pairing requires also there, and nucleosomes which functions in that. Okay, same thing is also found in the our body, that is hemoglobin that moves, that takes oxygens at various organs. That is also one of the functions of the protein. Now transport protein channel carrier proteins, as I said previously, in membrane structure also you will find same proteins that will transport it through various membrane structure. Another example is immunity antigen and antibodies, and this picture shows you how it functions. Now we will start with the another topics, that is physical and chemical properties of proteins. Whenever the physicals they are tasteless, colorless, odorless, crystalline compounds. Understand what is physical? Colorless, tasteless, odorless, crystalline compound. Now another that can also ask as a reason to amphoteric nature. Because of presence of amino and carboxyl groups, they can act as both acid and base and react with the acid in alkali to form salt. This property is known as amphoteric property. Now, isoelectric point. Protein can be or could be acidic or basic, positively charged or negatively charged. As I said, the pH at which the positive and negative charge are equal, then net charges become a zeros. The protein become a neutral. This pH is called isoelectric point or isoelectric pH for that proteins. Now, example of proteins: milk caseins, gelatin, hemoglobin, insulins. Okay, these are the. Now, another properties of chemical properties of the protein is salting in and salting out. Addition of reagent that make a protein dissolved in that reagent in process called salting in. And conversely, addition to any reagent make a protein precipitation. When the precipitation is there, then is process called a salting out, salting in and salting out. Now, hydrolysis. Hydro means water. Lysis means it breaks. Okay, so protein can be hydrolyzed by dilute mineral acid, alkali or proteolytic enzyme. The end product are amino acids. Protein plus water. That will gives you proteases and peptones. How it is formed? That is in our gastric pep juice, that is pepsin enzymes. Now these proteases or protein or peptones that will hydrolyze with the water, it gives you a peptides, small peptides, that is in intestine. That is enzyme called as intestinal trypsin. Okay, now these peptides further hydrolyze with the waters, that will gives you a intestinal trypsin, and this intestinal trypsin gives you a amino acid. This is absorbed by the intestine. So how we get amino acid in our body? That is through these enzymes. Okay, now another things, these what we are studying, chemical properties of protein. Now, denaturation. Denaturation of protein can be possible by various ways. That can be from high heat, change in the pH, or treatment with the various chemical like urea. That can also be possible, the denaturation of the protein. So, the term native protein is denoted to biologically active protein having a natural quaternary structure. When protein are exposed to extreme environments such as high heat, change in pH or treatment with the urea, they lose their biological activity, which denote the denaturation. Now, how the denaturation forms in the biological properties called denaturation. The denaturation could be reversible or irreversible. Okay, so there is a change in the physical or chemical properties when the protein are denaturation. Now, same thing is with the coagulation also. It is one of the properties of the protein. During the process of coagulation, there is an aggregation of the colloids, molecules, and protein solution become opaque. Remember, it is not a denaturation as a protein. Remains biologically active when it is coagulated. Now, blood group typing and oxidation is, as you know, putrefaction or decay of the protein is the oxidation process, and there is a bed order and decay of the process. This is all about the protein properties. Okay. Now, another chemical properties, color reaction of the proteins. Their various taste is given: xanthoprotic, Million test, nitrocrusated, Mollis test, and lead acetate. They gives the various colors and presence of various amino acids can be detected through these. For example, Million test, that gives you a presence of tyrosine is there or not, presence of cysteine, that nitrocrucide test. These are the various tests. Test is also available. Okay. Now we will see